Kia ora koutou katoa, ko Mick Black taku ingoa. Nau mai, haere mai. Welcome to our Friday afternoon Genomics Aotearoa seminar series. I'm Mick Black. I'm one of the co-directors of GA. I'll be hosting today's seminar. And I didn't check this, but I think one of our presenters today is the most presenterist of all of our seminar series. I think, Suzanne, you have the distinction of having appeared in the most GA seminars. So, two? It was only two. I thought you'd done three. Oh, yeah, see, 2.5. I could be wrong. I know I've introduced you multiple times. So today I am delighted to introduce Suzanne Manning from the Public Health Agency, Matt Howes and Maria Hepi from the New Zealand Institute for Public Health and Forensic Science, which is a little bit of a mouthful, but it's not too bad. We'll get used to it. Thank you all for coming along. Today's talk is Tikanga Māori for Advanced Cell Systems Research. So I'm going to hand over to you guys to take us through it. Thank you very much.
Kia ora, thank you very much. So I will start with sharing my screen and start with a karakia. So actually we're all seeing this. Okay. Whakataka tahau ki te uru, whakataka tahau ki te tonga, ki e mā kini ki utu, ki e mā tara ki tai. E hi aki ana te ata kura, ki tio, ki huka, ki hauhi, ki hei mauri ora. So kia ora koutou, ko Suzanne Manning tōku ingoa, presenting with me today are Matt Howes and Maria Hepi. And we're going to talk to you about a project that we did that was internally funded with ESR, and it was done in 2023 and 2024. And we have been arranging to present this for, like a good 9 months to 12 months or so. So the branding here is ESR. So we decided to keep it like that rather than change it to Public Health and Forensics, because it just gives you an indication that this is a prior project, because as we work through it, Maria will update you with what's happened since the this project happened. So the project was Tikanga Māori for organ-on-a-chip research. Now this pioneer project is the ESR name for the smaller internal funding project. It ended in June 2024. It was a participatory action research project. By that we mean that the researchers were the team, the whole team were involved in researching on themselves. The objective was to develop guidelines for culturally sensitive practices in the lab. By that we mean Māori practices in the lab. And so that's a cute little picture of maybe a organ on a chip, that whether or not the chip's really, Matt's nodding, so I guess that actually looks like that. So my role and Maria's role was for the social systems team. We were working with the people in the lab who were working on organ-on-a-chip research, and Matt will be explaining all about what that was. So over to you, Matt.
Thanks, Suzanne. Kia ora, everyone. My name's Matt, and I'm a postdoctoral scientist at PHF Science. Getting used to that new name. So I participated in this research during my PhD, which I finished last year at Otago University, and that was in collaboration with then ESR, looking at some of these new organ chip models that were kind of coming of age during my project. So the model, I guess, that we started with looks a little bit like what Suzanne had on the previous slide, but it would have been, I guess, handled with someone with gloves on. And the pink and blue colors were just the clear cell culture media. So yeah, I'm going to give a quick background to the program, the Advanced Cell Systems program that's at PHF Science, and where it came from. So this kind of field of advanced cell systems, or there's a few kind of other terms that are used in the field, like complex in vitro models or microphysiological systems or organ-on-a-chip systems, kind of came about in the late 1990s, early 2000s, where there was an interest in trying to make cell culture models or in vitro cell culture models more realistic, so that some of the kind of questions around the relevance could be answered. And I guess the aim, kind of capitulated in this picture on the right-hand side of the slide, is to model one organ or a couple of organs, or I think originally they were aiming to model a whole human body in a lab in a better way than kind of static cell culture systems previously did. So this research really progressed kind of around 2010, where there was a paper that showed really promising results with a chip that could model the lung breathing, and it could apply stretch to kind of cell cultures. And this led to the forming of a company called Emulate, which really kind of still sit at the front of this field with really complex but customer-focused models where you can actually commercially, where you can buy them from Emulate. And that's what we've been using. So Jan Powell, who I'm standing in for today, she received an award from ESR to go and investigate some of these advanced cell systems and how they could be used in New Zealand's unique, I guess, health questions. And she did, I guess, a lot of looking around and trying to find out what new models were out there, who sells them, what they're good for, what they're not good for. And some of these types of systems that she looked at are listed here, such as organoids, spheroids, scaffold systems like transwell cell culture inserts, and the more complex organ-on-chip. So each of these have different advantages and disadvantages to each research question, and they can be combined in different combinations. But where this research really started was with the organ-on-a-chip systems and a single replicating in vitro cell culture line. And some of the supposed advantages of these systems are that we can reduce animal use if these models are more realistic. So there's some new FDA directives around adopting more complex in vitro models to reduce or complement more specific animal research. To be able to restore some of the 3D tissue architecture, just due to the nature of how these chips are made with quite complex architecture. And then to be more physiologically relevant, restoring some of those parameters that organs hold, such as stretch or peristaltic flow or vascular basolateral epithelial kind of layers. So with that, could I be turned to the next slide? Thank you. So that kind of sets the background for the Advanced Cell Systems group. And part of this was kind of starting to look at where we were using cells already and what we hoped to use in the future as part of this research, and realized quite quickly that we were already using replicating cell lines before this kind of Tikanga project was initiated. And I think this will be brought up by Suzanne and Maria, but a really good opportunity for everyone involved to pause and kind of think, what are we doing? What could we do better going through this project? So where we were sitting was definitely with commercial cell lines that we could purchase from overseas, from a few vendors pictured on the right, and there's some other ones now that we've expanded into buying cells from overseas. The hope, I guess, in the future would be that there was something New Zealand-based, but generally purchasing cells from overseas, I'm sure there's other people in this room that have experience with that as well, is the easier, more accessible way to go. And again, there's just advantages and disadvantages to each one. Some of these commercial vendors are horrendously expensive and rapidly kind of suck up most of the project budget, but they come as validated cells that we know will be viable in these different models over the time course, so we can actually answer our questions properly. And the other problem, I guess, would be the lack of some of the specialized equipment that these companies and groups have, particularly in America, to separate out these types of cells and package them up in a way that they're going to be able to be used. So again, this is Jan's slide, and I believe she kind of tells us as a story looking at some of the history around cell culture use and the story around Henrietta Lacks's cells that were isolated and one of the first, probably the first replicating cell line. And then I think Jan kind of draws some parallels to the research that we were doing or what we can learn from that. So it all comes under this banner of the promise of using human cells to advance human health and science, and the aspects of that and what it involves. So starting with kind of a respect for the human donors. If this is something that changes over time, and I don't know if anyone's read the book from The Immortal Life of Henrietta Lacks or seen the movie, but provides quite a sobering story, I guess, of respect for Henrietta and her life, as she had an aggressive form of cancer. Brings up ideas around informed consent and what that actually means, if people understand what we're talking about as scientists, or if the family understand what was happening to Henrietta. There was scenes where her family thought that it turned into some kind of Frankenstein, whereas actually it was cells that wouldn't live unless they were under a certain condition. I guess this has relevance in New Zealand as well. The changing standards over time. We were looking at manufacturers that isolated cells potentially 10 or 20, 30 years ago, and things would have changed since then, and changed in what the manufacturers were able to supply us in terms of whether informed consent was acquired. Some of the ethics, there's parallels as well. Who decides what we get to do with these cells, what direction the research goes, and who benefits from the person's donation often. If it's these larger companies that are able to sell them and make money, that's a bit of a question that we started to think about. And then always cultural sensitivity and safety, kind of underlining that with unique instances in New Zealand as well, as this kind of science sits alongside Te Ao Māori, and how this might be different in different locations in different countries. So I'll leave my bit of the talk there. Happy to jump in again if there's kind of sciencey things that need to be answered. But this is where we were sitting with kind of what we knew already, acknowledging that we could do better, and Suzanne and Maria will outline a bit more of the social science and the future of this project and work.
Got it, Matt. Thank you for that. So Matt's well outlined the ethical type of issues we were talking about. So when we were looking at the organ on a chip work developing more, we were aware that, you know, as public health and forensics that we are now, that public good for this work in Aotearoa, New Zealand, should mean looking at different responses of cells for different populations of New Zealanders. So that leads us to getting the aim of having Māori donors giving us cells that we can do this work on. And if we're doing that, how can we make sure that they and we are comfortable with our processes in the labs? But beyond that, just how are we in a country that still has Te Tiriti o Waitangi as a foundational document, how are we honouring that in our lab processes? So we started to look at this, and this was how we did it. So we had a timeline where we weren't going to rush it, that we were actually going to talk about it, but we had mostly online, because we were spread over like both motu, and we did fund three actual in-person hui, and the first one was where we looked at the concepts so that everybody in this team would have the same basic knowledge. Then we had online chats about things. We came together to discuss some guidelines. Then we went away and did more editing, and we finished up with a celebration. We contracted Dr. Hokimata Harwood as our Māori cultural advisor throughout this process. So she gave us some feedback. Now, here in the first meeting, the hui timatanga, it was introductions to people and project, and actually a lot of the people, we got forensics people, as well as the advanced cell systems people, as well as the social systems people, all in together. And it actually took a lot of introductions. That took a long time, but it was really very good, because if we were going to work together, we needed to be able to discuss the issues without getting worried we were offending everybody. So knowing each other was a very important thing. We looked at the Māori concepts in Te Ara Tika and other documents, and we moved forward. Now, the thing with using these documents, I think they're on the next page, yes. The basis for this project was that we had a team who were basically non-Māori. And it was a small project, and it was only one of the things that we were doing at ESR as a whole. So it's not the sort of thing, and there was going to have to be a lot of upskilling to basic knowledge. So these documents here, or the concepts in here, for some of the team was new knowledge. We did not want to take Māori resource to educate Pākehā. We did think that it was our responsibility to do that work rather than call on Māori resource. Not to mention that the people who know about this sort of things are heavily overcommitted. Everybody wants their advice. So we took the approach that these three documents, Te Ara Tika, Te Mata Ira, and He Tangata Keitua, had been written by Māori experts workshopping. They had been written for non-Māori researchers, and there was a lot of good information with this. We took these documents and the knowledge that some of the team had to work together as a group to make sure that, to see what we could use these for. Part of the project, therefore, was to see whether this was a feasible approach. Were these documents, with some guidance from within the team and our cultural advisor, was that sufficient to come up with something? And a short answer, we think it was. Everything can be refined, but it was a useful process. So that's a good thing. Now, this is basically the sort of concepts and the sort of questions that are in Te Ara Tika and the other documents. So the four po of whakapapa, tika, manakitanga, and mana, which in these documents have quite a specific meaning. And the questions that are here are basically around the sort of questions that were in those documents, but we had put this up here for our project in particular. So we were talking about the history, consent in the context of cell lines. How do we feel about spirituality aspects? That was a thing. We were talking about how do you feel when we do karakia? And one of the things of having that karakia at the beginning, it became quite a robust discussion about not only why we would do such a thing and what we would do, but how people feel about doing that, given their own personal backgrounds. So the benefits of discussing this in the team were quite significant. In amongst the concepts that were in those documents, Te Ara Tika and others, the takoha was the aspect that we focused on most, especially in biobanking sort of idea with the donation of cells. We're talking about a gift, but it is a gift that has tapu associated with it. That is, it's conditional. It is restricted. And so there were a lot of issues here, as shown. That diagram is straight out of one of those documents, if not more than them. And we looked at the idea that the gift carried responsibilities, and we as a team were looking at how we could meet those responsibilities. What responsibilities do we have towards the human cells we work with, in particular recognizing the people that are behind those human cells? So this framework was what we focused our discussion around, again, from the documents. So there is this sharing the gift, using it in the spirit of the gift, and returning the gift. So we tended to look at both the spiritual and the physical level for each of these aspects, recognizing that the consent process, when somebody donates cells, is not just a tick box, but it actually creates a relationship between them and us, the researchers. It is both a physical and spiritual relationship. So we're looking at ways we could honour that. The expectations around the use of the gift, what is it appropriate to do, and what should we not do, and how do we know, and where's our level of comfort with that, and who decides? How do we decide? These are questions we were asking ourselves. So and returning the gift, and we quickly discovered that it was logistical impossibility, regulatory impossibility to return the cells themselves. So what is it that we would be returning, and how would we return it? Because that closing the loop, the reciprocal nature of that is a very important part of this whole tikanga. So our central question, after we had done our first week and we had understood the concepts, we went into discussing what it was going to mean for the research. And this is what our discussions sort of looked like. We had weekly half-hour team meetings, and people were really good at actually coming each week. And we used a meta board that was there, people could look at and come into it in between our weekly meetings. We discussed various things with relating to the framework that I've just shown you, and some of that was put onto the board. As people discuss things, the following week, I would reorganize the board a little bit, other people would too, and do those connections, but also try and pick up on the unanswered questions from the last week and use that as a springboard for the next week. And sometimes I would be worried that we didn't have enough to really go on. We only had sort of one small question left over from last week, but almost inevitably, by the time we finished the next session, I had 3 more questions, plus we'd covered a whole lot. So people really participated, and we did notice how everybody participated, from students, lab techs, science leaders, from the forensics, as well as the advanced cell systems and social systems people. Well, you don't normally have problems getting us talking. Grace Deng, a student, took photos, which was really great for some of us who didn't actually know what the lab looked like. because a lot of what we were talking about was the physical handling of the cells, and an idea of separating from, for example, the animal cells that we might be using in the lab, and what was physically possible. So here are some of the things that we had. The biological hood, the Zoe modules, one of them, two of them. That's what a Zoe looks like. Got two little pod type things, and there's a few of them in there, and a chip that has two channels. And if you have any questions on this, Matt is your person and not me. But for us, this was really useful, and it was really good that Grace came back and she did this because she thought we needed it, and we did. It was great. So then we had our second meeting. I drafted this from the discussions that we'd had and where we'd landed to. By the time we got to March, we were at a point where a lot of things were becoming clearer. So we wrote guidelines, a draft guidelines, but as you can see, there were comments, there was, highlighted things for discussions, they were all over the place. So the draft did get pulled to pieces quite a bit, which is great. And that's what we did at the second meeting. We did talk about a few things and what was possible, what was doable, and how we felt about it, because that is actually a big thing if you're going to get it, if you're going to have anything that's sustainable. This basically the things that we came up with, we made it simple in the end. We did a lot of discussion and we came down to only a few things really. So we talked a lot about consenting processes and making sure that we documented things well, but that individual donors that we could identify for research purposes was a whole future piece of work, and actually Maria is working on that now. When we received karakia, the physical part, physical, spiritual, when we physically received cells in the lab, we decided that we would have a karakia to clear the path for it. With the spirit of the gift, we were going to separate human and animal cells in the lab. We were going to ensure appropriate research questions so that we weren't wasting the gift on trivial questions. So returning the gift, people we thought would be giving the gift of their cells so that we could improve things in the health line, so that we could have scientific impact. And so it was important for us to do that. We decided that to collectively acknowledge and show gratitude for the donors, an annual hui would be a good way to do it, rather than trying to be individualized, because it's very difficult to do that sort of a thing. But you'll notice that these things, some of which are that we were looking to write lab guidelines, but some of these are not the lab team that will be doing it. Some of these are the bigger questions, like ensuring appropriate research questions. That's a much wider program level, if not a group level, where you might get the chief scientist involved, even in terms of setting some of the strategic direction. Annual hui, would you do it just for this sort of work, or is it actually, as Matt alluded to, there are many other, or there are other programs that use human cells, so maybe we should be doing it bigger. So we were not quite sure at where we're going to leave this, which it is a thing for moving on. So we had our final meeting, we celebrated reaching the end, and it was a nice little tidy thing that we've been talking about with people. And so here are the people who were involved in this. It was quite a collaborative effort. And then the next step, we were going to be implementing it and also working on this idea of the future where we would move beyond commercial cells. Now that was, as I say, middle of last year, and things have been moving on. So I will now hand over to Maria, who has been working on the next steps in terms of the social science part of it. So Maria, I will stop sharing so that you can see Maria in all of her glory.
Kia ora koutou. My internet is not that great, so hopefully it doesn't cut out while I'm talking. And I haven't got a lot to say, but we did get 3 more years funding through SSIF internal funding, and Matt's working on the science side of that project as well. So what we did, we had these guidelines, but then we were really thinking about what happens when we are working with people that are giving their cells from within New Zealand, especially for Māori. So went and talked to Maurice Curtis up in Auckland University's in the Brain Bank, and they've kind of been through this process themselves. They kind of started off with some guidelines, but then realised for them it was more about the relationship that they were having. They had kind of like a kahui group of kaumātua that they talked to that helped them develop the guidelines, and it was more about that relationship with them, so that when things happened in the lab or they had to do something, that they could just pick up the phone and just say, what do we do here? So, and through that, and also looking at a lot of the literature, we've kind of come to the realization that we need to move further than guidelines. What we do need is a kahui group, whether that be kaumātua or experts in this type of area. It can't be, but the thing is, it's all about funding. Who's going to fund those people? And it can't be by project. So, because it's about that relationship with the people that you're working with. So I have discussed it with the GM of Māori Impact, and he agrees, and he's right, yep, that's got to be organisational led. So because as Suzanne alluded to before, we've also got the DNA forensics, the DNA bank for forensics. We also do wastewater testing, and that involves human DNA as well. So yeah, it's kind of like looking towards the future, and really we do need to start looking at how we are going to do this and who do we need to work with to guide us on this journey, because it is really a journey. So that's kind of where we are now. So it's kind of trying to get the organisation to agree to fund outsiders to support us in this journey. Kia ora, and I'm glad the internet didn't cut out.
Oh, no, it was great. Thank you, Maria. Because it speaks to the funding system and what we get funding for, how much core funding, how much we can put this sort of thing within projects, but we don't need it project-based, it needs to be organisational-based. And Maurice, particularly his view was guidelines was a good start, but they had matured. They had moved beyond that. It's like you might have a written agreement with somebody, an MOU, but after a while you just work together, which is the ideal. So we've identified here the vision for the future. We just have to get the roadmap to get there. So kia ora koutou, we are available for questions.